Hello everyone. I'm Eric. And today we're gonna talk about what we did last year as an iGem team. So I'm one of the members who did uh who did research uh in the wet lab section. Yes. So our topic was about like the vital implementations of shRNA on novel CRC treatments, and I will just cover the basics. So first of all, so this is a really important uh this is a really important concept that is involved in our research, which is RNAi. Um, so the full name is RNA interference. This is a process which is highly conservative within any living organism. It is present in no matter like in this case the C. elegans or in our case the Homo sapien. So this system was first discovered by Andrew Fire and Craig Mello in 1998. They published a paper in 1998 and won the Nobel Prize in 2006. So they discovered this first in C. elegans. Um, this was this was a breaking discovery in cell regulation, where the where it mainly discusses where it mainly uh, explained how the body and how the cell regulated genetically regulated uh, regulated expressions. So this process involves siRNA, miRNA or shRNA being transfected or transcripted within the cell. It joins with the dicer protein and then with the RISC complex, and then targets the mRNA to do post-transcriptional regulations. So this is the diagram. So here, this could be this starting section of a DNA or of a RNA could be any in any form. Like in this case, we used a plasmid. Then it is cleaved by the dicer protein. Then it assembles with the RSE complex, gets activated, and targets the mRNA. So this is a post-transcriptional regulation because it halts translation, as you know, like the central dogma. So after transcription, there's modifications, and this is the regulation that is carried out to prevent translation, which then silence the gene. So this is one of the epigenetic uh, regulation systems within the body that regulates gene expression, and this is the process. So here is the mi, si, and shRNA. So miRNA stands for microRNA, siRNA stands for small interference RNA, and shRNA stands for small hairpin RNA. So probably you've heard of that miRNA is won the Nobel Prize in 2024, although they published the paper a long time ago. So these are all forms of non-coding RNA. Furthermore, like some examples such as the tRNA in translation and long non-coding RNA, which is also some related post-transcriptional regulation RNA. So it's normally 21 to 23 base pairs long. And in this case, the miRNA equals the shRNA. The only difference here is that the mRNA is generated within the cell. So it's the cell regulating the gene expression, which is what the whole thing is about. So we utilize this method to regulate genetic expression as how we want, but the cell also does that to regulate it based on the state that it's in. So this is the mRNA diagram. So as you can see, a gene has been transcripted and forms a miRNA, and it joins the RNAi, like this RNAi degradation system. And this is like what I described previously of how the cell regulates its genes by itself. Yes. So this is the cancer that we target, which is the colorectal cancer. So this is the main approach that we have taken. So for its anatomy, there's a few kinds. There's few kinds of colorectal cancer, which is in the transverse, descending, sigmoid, and rectum. So if you learn your anatomy, you can you know that it's in different sections of the intestine. So colorectal cancer is grouped, so colon cancer and rectal cancer, they're all grouped together due to their similar characteristics, um, like on a cellular basis. So, we target colorectal cancer as a whole. So this is our approach taken, which is the Hippo pathway. So this is one of the core pathways, obviously, that's really famous in the current years, in the research. It's really like emphasized on research these recent years, but it's one of the main pathways that regulates proliferation and apoptosis. So the core transcription factor here is the T84 protein, which is the TA transcription domain 4. So this pathway is first discovered in 2003 within Drosophilias. As you can see above, this is the structure of the pathway in Drosophilias and below in Homo sapiens. So the downstream gene controlled is responsible for proliferation and development. So thus, if there's an oncogenic mutation, there's some thing, there's some mutations that has happened within this pathway. So as you can see here in this diagram, this is the whole structure of the pathway with four upstream kinase cascades and the core proteins. So the YAP is the core transcriptional coactivator called the yes-associated protein. These are the two important kinase involved within this pathway called MST 1/2 and last 1/2. Um, so these four factors affects and controls the Hippo pathway, and including the cell cycle. So if during a cell cycle, during the specific stage, you want the cell to divide, so it activates the CDK8 protein in a yet unknown way, which uh, activates or inhibits like the Hippo pathway. So there's so as I mentioned before, there's an oncogenic mutation within a section or a portion of this pathway that leads to the upregulation of the apoptosis and proliferation gene. So the significance of this pathway, or like the T84 protein that we targeted, so there's a mutation of this T84 protein. As mentioned in a research, I think I remember published in 2016, T84 is shown to be upregulated within cancer cells. So there must be some oncogenic mutations suggested that if it's suppressed, the goal of suppressing cancer could be achieved. So we used so our method and procedure is to focus on the implementations on the theoretical side. We used a database called InVivoGene, or like made by InVivoGene called the siRNA Wizard, to design our siRNA sequence, and this is furthermore synthesized. And we use SW480, which is one of the non-metastatic CRC cells. So SW680 is the metastatic stage of the CRC cell, and SW480 is the non-metastatic stage. So we used that as a form of visual simulation. So we measured the ROS cell counting kit 8 level, which is used to measure cell viability, and the transwell level. These all measure like the cancer characteristics So the cell counting kit 8 experiment, this is the procedure. Um, I don't really need to read it, uh, but this basically it's the process of using this chemical and um, in a viable cell, there's metabolic processes, and these like NADPH, for example, will transform and reduce this WST8 into this formazine WST8 formazine, orange color, which is then shown, which is then measured quantitatively through a machine that measures like the intensity. And this is the results. I didn't include the data here, but uh, as you can see, the cell viability has decreased, I believe. Oh wait, no. So this cell viability has increased because using a formula that we have. But here, the orange color has decreased significantly. So this is the blank plasmid that we transfected. So as you can see, there's no result. But after the transfection of the two shRNAs that we generated, there's a increase in cell viability, which means the loss of cancerous characteristics. So the transwell test, as seen here, um, is also a way to measure the cancer migration. So there's this cancer. So this is the diagram here. We have a, a little compartment here. Um, this is the part where the cancer cells are initially placed, and there's this uh, membrane with pores through it, and the cancer cells can migrate through these pores to the lower compartments because there are important nutrients that they need. That's also a chemical bait, basically. And we count the number of cancer cells that migrated through these pores through a specific app called ImageJ. So this is the data here. As we can see, less migration. There's less migration of the cancer cells after the transfection of the shRNA. Um, so as you can see here, the shRNA is 2 is more effective than shRNA 1. So this might be due to many reasons, such as yet just unresearched reasons, such as how which segment of targeted mRNA sequence might affect the results. This is yet unresearched, but uh, shRNA1 and shRNA2 they target different places within the within this within the mRNA, thus achieving different results. So ROS stands for reactive oxygen species, which are metabolic byproducts that are normally degraded. However, in cancer cells, due to metabolic reprogramming, um, there's uh, there's an increase in RS levels. Um, uh, and basically there's a metabolic change. So here after the transfection of the shRNA, we measured the RS level. And here, as we can see, there's a increase of RS level, which suggests that there's a loss of cancerous characteristics metabolically. So is mRNA like dead? Like this is a part like is mRNA and related therapeutics such as siRNA therapeutics dead? Just keep it at the back of your mind and think of it. So this is the improvement. Think of it just now. Okay. So this is an improvement experiment. Um, we created a mutated form uh of T84 um with as you can see, the 105 to 109 is deleted. So this mutated form of the protein is like inserted into the cell, and like somehow in and yet unobserved in and yet unresearched uh, phenomena that we observed is that like surprisingly it also achieved like the results or even the better results compared to shRNA. This is yet unresearched, so uh, I cannot explain it molecularly, but this is probably due to uh, some relations that interferes with the original T84. So again, NLS here stands for the nuclear localization sequence, which is an amino acid sequence that gets involved in this nuclear localization process, where a transcription factor is introduced into the nucleus for regulation. So here we removed it, and somehow it interferes with the original T84. So this is our research that we conducted. And I must mention the flaws because all what we did is vitro simulations, right? If we want to bring it to therapeutics, we have to study the vivo implementations. Like we have to do vivo experiments. Um, uh, and for example, one of the disadvantages of mRNA or like siRNA uh, vaccine is that it's easily degraded within the body. So there must be some effective ways that regulates the mRNA to not be degraded, which I'll mention later. But this is a problem, right? If we want to make it into a therapeutics, it must be solved. So this is a really interesting topic posted by a team on Nature in 2023, I believe. So they use this also. Uh, they This is the YAP, which is the core transcriptional coactivator. So what we did in the NLS is target the core transcriptional factor, and YAP is the core transcriptional coactivator. So they target the SOX9 protein, which is core in the YAP YAP process of nuclear localization. So they created this helix that interferes, that binds with this SOX9 protein, which uh, which uh, the YAP has no SOX9 protein to bind, in easy words. So it cannot enter the nucleus, uh, thus achieving regulation. Again, what we did is what we can do yet, but there, as you can see, this complex structure of the Hippo pathway, there's a lot more to explore. So at last, I want to leave it as a really I'll try to inspire one of you. So I don't know what you guys will do for this year's topic, but what I'm thinking is that since everything is so complicated, and we just contributed a bit, why not just think? Why not just meditate over the other possible uh, researches that we could do, right? So as you can see, this complex structure here, there's not just one way to regulate, right? We can, for example, limit the energy the cancer gets in specific ways that doesn't that specifically targets cancer cells. And that's what a lot of researchers are doing nowadays. So what I want to suggest here is that there's not only one method, right? There's this complex cell biology, can't like oncology, that there's too much to do to there's too much about oncology, and I really want to tell you guys to do your research, read your research papers, and think and meditate, really meditate over what you can contribute to the world. So this is my answer to the question, is mRNA therapeutics dead? I would rather say largely, but there's new effective ways, such as this discussed in this epigenic conference, that there is this way of generating this virus-like structure within the body, which protects it from being degraded. So again, that's it. I just want to tell you guys that there's a lot much to do to it. And again, I've been continuing to brainstorm over the other topics that we can that I can do as a student, and try to do it yourself. Okay, thank you.